Hello, thank you so much. I'm excited to be here today to tell you about an interesting phenomenon that we observed while making virus-like particles for SARS-CoV-2. And we think that this can serve as an interesting new reporter system for particle production. So, virus-like particles, or VLPs, have a long history in the field of virology with many uses across different types of viruses. And this is because they're really easy to use, they're simple, they're very safe, and they have widespread applications as vaccines or as reporters for entry or assembly. So for SARS-CoV-2, we make VLPs by overexpressing the 4 structural proteins, the S, E, M, and N proteins. And in cells, these have the expected localization, and upon co-expression, they aggregate on cellular membranes and then bud from cells. So we devised a system to purify these budded and secreted particles by a density ultracentrifugation of a density step gradient. And so we can purify each fraction of the gradient, and then with mass spec and Western blotting, we can identify viral structural proteins in various fractions of the gradient. We can then compare these to purified virus, and we can note that the VLPs are extremely much more heterogeneous than wild-type virus, but we do have an enrichment of viral structural proteins in the lower density fractions of the gradient compared to the higher density fractions found in the virus. So we wanted to turn these VLPs into a reporter system to measure VLP entry into new cells. And so we, of course, wanted to use a reporter RNA. And the best way to do this is by using a reporter RNA and attaching it with a packaging signal to a structural protein. And we chose to use a well-known, established reporter RNA from the bacteriophage MS2, and so this has an established packaging signal that binds very tightly to the capsid protein of MS2. So we tethered the MS2 coprotein to the SARS-CoV-2 nucleocapsid protein, and then generated a reporter RNA with the well-known stem loops from the MS2 that bind to the coprotein. So then we reasoned that overexpressing these in conjunction with the SARS-CoV-2 structural proteins would generate VLPs that package a reporter RNA. To test this, we collected the supernatant from VLP-producing cells and with QRT-PCR measured our reporter RNA, and we found the secretion of our reporter RNA in the supernatant, as expected. We reasoned, though, that any RNA that is actually packaged into the virion, or into the VLP would be protected from RNase A degradation. And so with an RNase A treatment, we then demonstrated that that our reporter RNA is actually packaged inside a VLP and protected from RNase degradation in a manner dependent on the MS2 coprotein. So then we added these VLPs to naive cells, and we found that across 4 different cell lines that are susceptible, we found nanoluciferase signal. So we used a nanoluciferase reporter RNA in this case, and then found strong nanoluciferase signal in a VLP-dependent manner. So we wanted to further characterize these VLPs and this pseudo-infectivity that we observed here, and to measure the efficiency of it. So to do this, we again added our reporter VLPs to naive cells, and then in conjunction, we purified the RNA from the VLPs, and using traditional transfection methods, we transiently transfected the RNA into naive cells. And in both cases, we observe that the nanoluciferase signal is in the naive cells. So this demonstrates then that the transfected RNA is competent in the naive cells. However, as you can see, there's a huge difference between the pseudo-infected VLPs and then the transfected RNA, indicating that our VLPs are so much better than traditional transfection methods. And we were quite curious about this. This was perplexing, and so we initiated a variety of controls to try to identify what the difference between these 2 samples are. And the clue to the difference between these 2 samples came when we finally inhibited cellular translation. So we found that when we added our VLPs to naive cells, and then at the same time added inhibitors of cellular translation, we found actually no difference in nanoluciferase signal in the naive cells. So this indicated then to us that our nanoluciferase reporter RNA is not responsible for the majority of the nanoluciferase signal that we're measuring in this case. And so that led us to hypothesize then that it must be nanoluciferase protein that is delivered to naive cells. And so that implies then that nanoluciferase protein is actually packaged in the VLP producing cells and then delivered to these naive cells. So to test this hypothesis, we took our nanoluciferase expressing cells and then overexpressed either our VLPs or a GFP control, collected the supernatant, and we found in fact that nanoluciferase is enriched in our VLP producing cells in the supernatant. And then when we further purified the supernatant through a sucrose cushion, we found again an enrichment of nanoluciferase, and in this case in a higher density molecular species in the pellet of a sucrose cushion. So this, of course, implies that nanoluciferase is in a large molecular species, and we hypothesized that this could indicate that nanoluciferase is actually packaged inside of the VLP. So to test this, we used a protease protection assay. And so here we treated our VLPs with PK to degrade any free nanoluciferase, and then simultaneously disrupted the VLPs with Triton in order to liberate any nanoluciferase to be degraded. And we found very strong protection of nanoluciferase in the VLPs with PK treatment. But when we disrupted the VLPs, we found a reduction in nanoluciferase signal. So this indicates then that nanoluciferase is likely packaged inside of a VLP. And we wondered, does this happen in infected cells? So when SARS-CoV-2 infects a cell, can it package a free cellular cytoplasmic protein? So to do this, we again took our nanoluciferase expressing cells and infected them with SARS-CoV-2 and collected the supernatant, and we found a virus-dependent enrichment in secreted nanoluciferase. And then we subjected these samples to our protease K treatment, protection assay, and again we see protection of our nanoluciferase signal despite protease K treatment, and then we see that we can remove this signal when we treat with Triton. So then we wanted to see, is this a general mechanism? Do other enveloped viruses also package free cytoplasmic proteins? So we used our two favorite flavivirus viruses and infected our nanoluciferase cells, and we see that, in fact, we do not see an enrichment of nanoluciferase secreted from flavivirus-infected cells. So if we think about some of the many, many differences between coronaviruses and flaviviruses, something that's very striking is the difference in particle size. So coronaviruses are nearly 3 times bigger than flaviviruses, and so we hypothesized then that perhaps SARS-CoV-2 packages nanoluciferase in a size-dependent manner. And so to address this question, we use 3 different luciferase constructs that come in different sizes that can be used then as a molecular ladder to measure the packaging of cellular cytoplasmic proteins into coronavirus. And so again, we expressed our 3 different luciferase constructs in cells and infected them or overexpressed our VLPs in the cells, and then measured the PK-resistant luciferase that is secreted. And again, with nanoluciferase, we see the protection of secreted nanoluciferase upon SARS-CoV-2 infection, and then interestingly, with ranililuciferase, we also see the secretion in a virus-dependent manner and its protection from degradation. However, with the much larger firefly luciferase, we do not see a specific packaging or protection of the firefly luciferase. And I don't have time to show you all the data today, but we also performed this experiment with the VLPs, and interestingly, we see that while nanoluciferase is again packaged and protected from degradation with the VLP system, we do not see the secretion or packaging of ranililuciferase with the VLPs. So this indicates then that while VLPs are an amazing system and very useful, they don't fully recapitulate virion assembly, and this makes sense with the gradients that I showed you earlier, suggesting that they're in low density fractions. However, the conservation though of packaging and secretion of nanoluciferase in both VLPs and virus, we think can serve as a very nice reporter system to measure particle secretion. So to test this as a reporter assay, we then took our nanoluciferase expressing cells and infected them with SARS-CoV-2, and then used an siRNA to inhibit to knock down the RNA-dependent RNA polymerase, and then collected the supernatant. And we tested for infectious virus in the supernatant as well as viral genome copies. And with both, as expected, we see a decrease in infectious virus secreted and RNA copies. And then we also see a decrease in the secreted and protease K protected nanoluciferase secreted from these cells, and so upon RDRP inhibition. So we think that this then can serve as a nice marker for virus particle production in a very simple manner. So today we've gone over and demonstrated that SARS-CoV-2 virus and VLPs package a free cellular protein such as nanoluciferase. We think that this encapsulation and secretion is dependent on the size of the protein that is packaged. Since we do not see this with the large firefly luciferase, and nor do we see nanoluciferase packaging with flaviviruses. So we think this can be a useful tool. And we're initiating some experiments then to look at what is the function of packaging cellular proteins into a virus, and then what sort of mechanisms might be involved in the VLP versus the virus. So with that, thank you so much to Judith Friedman, my PI, and members of the Friedman Lab for their support in this project, and to people in the Andino Lab for reagents and their support. And thank you so much for your attention and the opportunity to speak today.
Thanks, that was really interesting. Do you have a sense if this is specific, where it's nanoluciferase kind of specifically? You're kind of saying that, oh, the size matters, but do we find other host proteins in SARS-CoV-2 VLPs? And did you try anything else? Did you, yeah, what do you know about the mechanism?
Yeah, we don't know much about the mechanism at this point. And I think, yeah, we are starting experiments to look at what other proteins are packaged. And I think there probably are other proteins packaged. But at this point, we have some systems designed, but we don't really know a whole lot yet. So.
Less behind.
Hey, thanks so much for a great talk. This is Syed from the Gladstone Institutes. Yeah, very similar to the previous question, do you think there could be a specific interaction or a specific binding with any of the structural proteins, or do you think it's just based on volume, like VLPs maybe have more free volume that is just passively incorporating?
Yeah, I think, so in this case, I do kind of envision this as sort of like a bulk packaging of just the cytoplasm. And nanoluciferase is abundantly expressed when you overexpress it, and so there's a lot of it there, and it's a free protein. It's not in complex with any other cellular proteins, and so I think that it is just kind of bulk packaged into the virus or VLP, which have a lot of space inside of them. But that definitely doesn't rule out that there could be specific packaging of certain cellular proteins, and yeah, but who knows?
Hi, so your flavivirus work does suggest that size does matter, but have you also considered stability? Because Nanonuc is horribly stable there is no half-life, and Renilla is about 12 hours, whereas Firefly is about 120 minutes. So potentially you could put a pest sequence on your Nanonuc and see if stability is also a potential factor in why you're seeing it.
Yeah, that's a good idea. I haven't thought a ton about that, but yeah, we could degrade it and see if that prevents it from being packaged. Yeah.
That's so cool. So, Carla Kierkegaard, Stanford. So your Nanonuc is being packaged. Presumably it's active catalytically within the virion, so you don't need cell entry for it to be active. Mm-hmm. So it's a packaging assay, but not an entry assay. Can you think of how to turn it into an entry assay as well?
Yeah, we are starting some experiments to see if we can use it as an entry assay for virus, and it could have a really nice comparison assay that way.
Or your VLPs, too.
Or for the VLPs. I think at this point, I don't know that the entry assay is very specific, but it does enter cells. It does enter naive cells and is still active in the naive cells.
Yeah, I wondered if you could package an inhibitor of the fluorescence or something in the virion, and then when it enters a cell, it dilutes.
Yeah, that would be a cool idea, yeah. Thank you.
Bob Siegel, Stanford. This is a follow-up on some of the other questions. So your comparator virus was actually an icosahedral virus, which has a very different assembly. I'm wondering if you've looked at, for instance, flu or another helical virus, which has a lot more, similarity to corona.
I have not looked at any other viruses, but yeah, it'd be nice to have some other size distributions, and of course, yeah, size is not the only difference between these viruses, but the way they package is very different also. But yeah, it'd be good to include other controls.
Okay, thank you very much.
Thank you.